Hey guys, this is Malinki. Welcome back to my channel, Voice of Malinki. Today we will talk about structure and function of carbohydrates in one shot. And if you are new in my channel, please subscribe my channel. And if you like my video, please do like, comment and share my video. So carbohydrates are the most abundant biomolecule in nature. They are primarily composed of carbon, hydrogen and oxygen in 1:2:1 ratio. That means 1 carbon, 2 hydrogen and 1 oxygen. Carbohydrates are also referred to as saccharides. It arises from the Greek word saccharin, which means sugar. Function. Carbohydrates are main source of energy for all organisms. Carbohydrates are precursors for many organic compounds such as amino acids and fats. Carbohydrates are structural materials. For example, carbohydrates such as glycoproteins and glycolipids participate in the structure of cell membrane. Cellulose and peptidoglycan are the structural units of plant and bacterial cell wall simultaneously. Chitin is the structural material in exoskeleton of some insects. Ribose is the building block of DNA. Carbohydrates such as starch in plant and glycogen in animal are the storage form of energy. Classification. So carbohydrates are classified into 3 groups: monosaccharide, oligosaccharide and polysaccharide. Monosaccharides. So they are composed of only one sugar unit. They are also referred to as simple sugar. Example: glucose, galactose, fructose. Let's see their structure. So glucose, galactose, fructose, these are monosaccharides. Now, based on the presence of functional groups, monosaccharides are further divided into 2 groups: aldose and ketose. When the functional group is aldehyde, they are known as aldose. Aldehyde means this is CHO. Example: glucose, galactose. So you can see here in glucose and galactose, aldehyde group CHO are present. So they are aldose. Now when functional group is ketone, that is CO, they are known as ketose. Example: fructose. So if we see the structure of fructose, we can see the keto group CO is present here. Based on number of carbon atoms, monosaccharides are divided into different categories such as triose, 3 carbon compounds, tetrose, 4 carbon compounds, pentose, 5 carbon compounds, hexose, 6 carbon compounds, heptose, 7 carbon compounds. So we will see their structures now. So triose aldose is glyceraldehyde. It has 3 carbon. Triose ketose is dihydroxyacetone, 3 carbon. Tetrose aldose is erythrose, 4 carbon. Tetrose ketose is erythrulose, 4 carbon compound. Pentose aldose is ribose, that is 5 carbon compound. Pentose ketose is ribulose, so this is 5 carbon compound. Hexose aldose is glucose, that is 6 carbon compound. We have seen glucose, and hexose ketose is fructose. Heptose aldose is glucoheptose, that is 7 carbon compound, and heptose ketose is pseudoheptulose, that is 7 carbon compound. So we have seen the structures here. Okay. So glucose comes from the food and our body uses it for energy. So glucose contains 6 carbon atoms, 12 hydrogen atoms and 6 oxygen atoms. Now, like glucose, fructose also contains 6 carbon atoms, 12 hydrogen atoms and 6 oxygen atoms. Fructose is known as fruit sugar because it occurs primarily in many fruits. It also occurs naturally in other plant foods such as honey, vegetables, sugar beets, sugarcane, etc. Galactose also contains 6 carbon atoms, 12 hydrogen atoms and 6 oxygen atoms. It is found in dairy products. Now we will talk something about D and L isomers. So D and L isomers are mirror images of each other. So let's see the picture. The orientation of hydrogen and wedge group on the carbon atom, specifically on this carbon atom, determines whether the sugar is D or L isomers. If the OH group is on the right side, the sugar is D isomer, and if it is on the left side, the sugar is L isomer. So we can see in D-glucose, in D-galactose, in D-fructose, this OH group is in the right side. Whereas in L-glucose, in L-galactose and in L-fructose, the OH group is in the left side. Let's see something about anomers. So anomers are cyclic monosaccharides. And monosaccharides are of 2 types, alpha and beta. So let's see the structure first. Okay. So in glucose and galactose, in alpha configuration, hydroxyl group attached to the C1. So this hydroxyl group that is attached to the C1 and hydroxyl group that is attached to the C6. So this one. So they are placed on opposite sides of the ring, right? This OH, and this OH are placed on the opposite sides of the ring. So they are in the alpha configuration. Whereas in beta configuration, hydroxyl group that is attached to the C1, this one, and hydroxyl group attached to the C6. So this one and this one are placed on the same side of the ring, right? So this is in beta configuration. So in alpha configuration, C1 position contains the OH group and C6 position contains the OH group. So these two OH groups are placed in the opposite side, and in beta configuration, these two OH are placed in the same side. So now let's check the fructose. So in fructose, in alpha configuration, hydroxyl group attached to the C2 and hydroxyl group attached to the C6 are placed on opposite sides of the ring, whereas in beta configuration, hydroxyl group attached to the C2 and hydroxyl group attached to the C6 are placed on the same side of the ring. So in beta configuration, they are on the same side. In alpha configuration, they are in the opposite side. I hope this is clear for you till this part. Let's talk about disaccharides. So they are composed of two monosaccharide units held together by a glycosidic bond. Example: maltose, lactose, sucrose. Let's see their structure. Maltose, lactose, sucrose. Now disaccharides are of two types: reducing and non-reducing. Reducing disaccharides have free aldehyde or keto group. Example: maltose and lactose. Non-reducing disaccharides do not have free aldehyde or keto group. Example: sucrose. So in this picture you can understand this better. So in maltose and lactose, free aldehyde group is there. That is this group. So here CHO group is free here. In lactose also, this is CHO. This group is free here. So these two are reducing type. And in case of sucrose, you can see you will not get any free aldehyde or keto group. So this is non-reducing. This is because the aldehyde group that is present in the C1 of alpha glucose and the keto group that is present on the C2 of fructose. These two are combined here to form this glycosidic bond. So there is no any free end, free aldehyde or keto end. So sucrose is non-reducing. So we will see them now. First is maltose. So maltose is also known as malt sugar and primarily found in grains. Now maltose, it is composed of 2 alpha glucose units and they are held together by alpha 1,4 glycosidic bond. Because in this disaccharide, C1 of this alpha glucose and C4 of this alpha glucose are involved in the linking process. So, next is lactose. Lactose is also known as milk sugar since it is found in milk. It is composed of beta galactose and beta glucose unit held together by beta 1,4 glycosidic bond. In this disaccharide, C1 of beta galactose and C4 of beta glucose are involved in the linking process. Next is sucrose. So sucrose is more commonly known as cane sugar. They are mostly produced by sugarcane. It is composed of alpha glucose and beta fructose units held together by a glycosidic bond alpha 1 beta 2. In this disaccharide, C1 of alpha glucose and C2 of beta fructose are involved in the linking process. Now disaccharides form when 2 monosaccharides undergo a dehydration reaction or loss of water called condensation reaction. Similarly, 2 monosaccharides form when a disaccharide undergoes a rehydration, that is addition of water, that is called hydrolysis reaction. So in this picture you can see 2 alpha glucose molecules are combined to generate 1 maltose, and here 1 molecule of water is getting released. That's why this is dehydration reaction where 1 molecule of water is released and it is called condensation reaction. And here also you can see that if you add 1 molecule of water in maltose, it will be hydrolyzed and 2 alpha glucose units will be produced. So it is the rehydration of addition of water and this is called hydrolysis reaction. So next we will talk about polysaccharides. Polysaccharides are also known as glycans. They are most abundant carbohydrate found in food. They are composed of many monosaccharide units bound together by glycosidic linkage. Polysaccharides are of two types, homopolysaccharide and heteropolysaccharide. Both homo and heteropolysaccharides are linear, that is unbranched, and branched. Homopolysaccharides are made up of only a single type of monosaccharide. Example: starch, glycogen, cellulose. Heteropolysaccharides are made up of more than one type of monosaccharides. Example: hyaluronic acid, chondroitin 4-sulfate, heparin, etc. They are also called as glycosaminoglycans or GAG. Starch. So, starch is the storage form of carbohydrate in plants. Starch is found in rice, bread, and many staple foods. Let's see the structure of starch. So, it is composed of alpha glucose units because this is homopolysaccharides. So, number of alpha glucose units are combined here to form starch. Starch consists of two types of polymer chains known as amylose, that is unbranched. Amylose is unbranched, and amylopectin, that is branched. So, starch could be of two types, branched and unbranched. Amylose possesses a linear structure. This is unbranched linear structure with alpha 1,4 glycosidic bonds. Alpha 1,4 glycosidic bonds are here in amylose. Number of alpha glucose are combined here via alpha 1,4 linkage to form amylose. Amylopectin possesses a branched structure that we can see here. It has alpha 1,4 glycosidic linkage as well as alpha 1,6 glycosidic linkage at branch point. So, at branch point, it has alpha 1,6 glycosidic linkage and it also has alpha 1,4 glycosidic linkage in their linear form. So, this is starch. Next is glycogen. So, glycogen is the storage form of carbohydrate in animals. The structure of glycogen is very much similar to that of amylopectin with more number of branches. So, the structure is almost same as amylopectin. The glycogen has more number of branches, like in starch. In starch, the branch point occurs every 25 to 30 glucose units, whereas in glycogen, branch point occurs every 8 to 12 glucose units. That means in glycogen, it has more number of branches compared to amylopectin. Otherwise, the structure is almost same here. Cellulose. Cellulose occurs exclusively in plants. It is the constituent of fiber. Now, cellulose is composed of beta glucose units, because this is also homopolysaccharide, so it contains many beta glucose units linked by beta 1,4 glycosidic bonds. So in starch and glycogen we have seen alpha glucose units are there. In cellulose it has beta glucose units. Now cellulose cannot be digested by men due to lack of enzyme that cleaves this beta 1,4 glycosidic bonds. We men do not have those enzymes that can cleave beta glycosidic bonds. Therefore, we cannot digest cellulose. But herbivorous animals contain microorganisms in their gut which produce enzymes that can cleave these beta glycosidic bonds. Therefore, herbivorous animals can digest cellulose grasses. So, next is hyaluronic acid, that is heteropolysaccharide. So, hyaluronic acids are found in skin and connective tissues. They are composed of D-glucuronic acid and N-acetyl glucosamine. N means many, because many numbers of this unit is there, which contains D-glucuronic acid and N-acetyl glucosamine. Therefore, it is heteropolysaccharide. More than one type of monosaccharide is there. And here one acid group is attached, C++, and it also has the amine group. After hyaluronic acid, we will see chondroitin 4-sulfate. It is made up of D-glucuronic acid and N-acetyl glucosamine 4-sulfate. Again, N means many. So, many number of such units are present in chondroitin 4-sulfate. Here, the difference between hyaluronic acid and chondroitin 4-sulfate, you can see that one sulfate group is extra here. Other than this, the structure is same, only the sulfate group is extra here. That is not present in hyaluronic acid. And this chondroitin 4-sulfate is found in bones and cartilages. This is one more heteropolysaccharide. Heparin is one more heteropolysaccharide. It is an anticoagulant, that is, it prevents blood clotting. And it occurs in blood. Now, heparin is made up of D-glucuronate 2-sulfate and N-sulfoglucosamine 6-sulfate. So, here also one acid group is present, C++, and number of sulfate groups are present here along with the amine group.